And now we talk about something that might be very close to your heart, which is molecular machines, because for those who do not know, Dr. Tour is quite renowned in his work with molecular machines, specifically nanocars. And maybe Dr. Tour will tell us what a nanocar is, but he has he got the Honda Innovation Award for nanocars in 2005. The NSF Presidential Young Investigator Award, that was quite earlier in 1990. So he's been into innovation for quite long, and he has also in 2005 Tour's journal article, Directional Control in Thermally Driven Single Molecule Nanocars. Single molecule nanocars. So one molecule that behaves like a car, he's gonna explain this to us, was ranked the most accessed journal article by the American Chemical Society. So Dr. Tour, you know plenty about molecular machines. Can you tell us what's a nanocar, just to start with?
Sure. A nanocar is a single molecule car. We put in it four wheels and fully rotating axles. It has a chassis, and it can roll across a surface. They're so small we can park about 50,000 of them, across the diameter of a human hair, just across the
50,000.
Yeah. So you can only see this with extremely powerful microscopes, scanning tunneling microscopes.
Do they carry cargo, or they just move?
No, they can carry cargo. We have cargo carrying ones. We've made nano trains where we hook these together and make trains.
Wow.
We've made 6-wheel versions, 2-wheel versions, 3-wheel versions, so many different models we've made. And in fact, in 2017, we won the first international nanocar race, which was held in France. And we designed the car, built the car, and then it was driven by one of my colleagues from University of Graz in Austria.
How do you drive them?
You use the electric field gradient of a scanning tunneling microscope tip. And we had to go 150 nanometers around one pylon, another pylon, and then through some posts, some end posts there. And
so you need to navigate it.
Right. And we completed the race in 1.5 hours. And the next group behind us was the Swiss team, and they came in 5 hours behind us. And none of the other international teams was able to finish the race in 30 hours. So we really excelled them all in that. And then the next nanocar race, it should have been last year, but due to COVID, hopefully it'll be next year, again in France. And but we published a paper right after the race on how to build and how to race a fast nanocar, because there's two things. You have to build it right, and you have to race it in the right way. And so we elevated the whole world. You know, scientists don't hide what we do. We elevate the world through that publication. So now the competition is going to be much harder because they've seen how we do it. And so it's just going to be more competitive now.
And since you've now explained it, it's even more fascinating. So I'm saying that nanocars are fascinating, but I have been, fascinated by those things that God has created into us that are called motor proteins. Things like kinesins and dyneins. And for the audience, those are, those, funny robots that, like have legs and move inside our cells, and they move cargo around. So Dr. Tour, you said you needed hours to go through a racetrack of 150 nanometers. And how does that compare to the performance of a kinesin or a dynein? And I would really urge the audience, after seeing this, to go and look on the internet about what a kinesin and dynein do, and walking over those microtubules. So how does the performance of the best nanocar that you have created compare to the performance of those cars that are there in our cells?
So the kinesin, those biological ones don't really look like cars. They're much bigger than what we are. We're just a single molecule. Those kinesins are macromolecules. They're very large structures in comparison. I would say maybe more than 10,000 times larger. So we're talking at least 4 orders of magnitude larger, maybe even 5 orders of magnitude larger in length. And in molecular weight as well. So they're much smaller. Our nanocars, we've built many versions that have motors on them. They're called Faringa motors. We put them in, and these motors can spin. You shine a light on them, and the motors will spin at 3 million rotations per second, and they'll move the cars much faster than we were able to move it in the races. In the races, we couldn't use a motorized car. We had to use the electric field gradient of a tip to draw it. They wouldn't let us use our motorized cars. Our motorized cars are much faster than these other cars. So with motorized cars, we could probably complete a race like that, maybe even in a matter of minutes. How do they compare? I mean, there is no comparison. Kinesin is so much more complex. Kinesin carries huge cargoes with it. Kinesin doesn't need an external individual acting on it. What it does is it moves across a microtubule, and it moves across this microtubule by sensing the electric field gradients that are going through that microtubule, and it progresses it and carries it along to bring its cargo where it needs to bring it. Those are nature's nanomachines. They're bigger than our nanomachines, but they are so much more complex, so much more functional. They do much more work than what we are able to do, far more sophisticated. And I just stand in awe of God. He does amazing things.
So, Dr. Tour, when I just saw those videos, and the kinesin it just looks like a person. So it has those legs, and it's like having its arms like this, and it's just grabbing on something, a vesicle or something, and it's like moving like this, and it's walking on those microtubules. And the walking, when I just looked into the papers that describe the chemical reactions that causes it to walk, and ATP, ADP cycles, etc., and how it grabs with a microtubule, and that it has adapters. So depending on the kind of the cargo, you'll have the right kinesin, as if you have the truck that carries cement and the truck that carries glass, and it's the same truck and the same motor, but it has a different container to grab to the right cargo. And then I look into these papers that say evolution of kinesins, and what are you talking about, guys? So the question I have to you is it rational to propose that motors like this and their microtubules, and it happens that ATP is around and they use it to walk, and the cargo and the adapters, can all of this come by chance with any stretch of imagination?
Well, I think the suggestion is not that it comes by chance, but that it comes by evolution. And
Yeah.
so then you have to ask the question, give me some details on that. What do you mean by evolution? How would something like this come about? How would, tell me, and they'll say, well, one small change at a time. So how many changes would it take? How many changes would it take in DNA to make a series of enzymes to build a structure that can do that? And what's the probability of being able to build a structure? How does it know to build a structure like that? There's so many mysteries. And again, to use this term evolution, just throw it out there and just feel that now stops all argument. It doesn't stop anything. It only just begs the question. Give me more detail. Nobody knows. Nobody knows. These things are so utterly complex. And what we're talking about, Ahmad, is just a single portion of the functionality of a cell that has thousands upon thousands upon thousands of portions of functionality. And how does this one portion how does this work? Nobody knows.
Yeah, and the reason I use the word chance is that, yeah, they would say evolution and small changes, but then they say that the changes are essentially random mutation, and random is, a chance event. So from your work in motors and cars, do you put a car there and you wait for some chance to happen so that you have a better car, or maybe a car becomes a train?
No,
Is this possible at all?
that doesn't happen. Not in our lab. That doesn't happen. And it's just hard to fathom. And people will take working cells today and show their processes and somehow extrapolate this to the origin of life, and somehow extrapolate this to the functioning, the new function. So you can have, you can see, evolution is a slippery term. It's a slippery term because some people look at it in the context of making human beings. Other people look at it in the context of very small changes within a cell. There are very small changes within a cell, and if you want to call that evolution, fine. But then you have to define things for us. So what these kinesin walkers are, these are amazing new systems. We don't have examples of evolution of a new system, of a kinesin walker from something that wasn't a kinesin walker. We don't have examples of that. We don't have examples of systems changing. We have examples within a cell of small changes occurring within an organism, small changes, but never a system level change. And this is what I'm talking about.
I noticed when I traced those kinesins back that they are in eukaryotes, which is like cells like animals and plants, but they're not there in prokaryotes, so they're not in bacteria, for example. And then I need somebody to explain to me how can the whole complete system just come out of nowhere into the first, least or the last eukaryotic common ancestor or something, which is an imaginary hypothetical creature. And there is no answer. They would just say it evolved, or it, co-evolved with something. And some people would just take this as an answer. And I had a complete debate with an evolutionary biologist, and I had this as my initial thesis. And he said, I'll bring you papers about the evolution of kinesins. And it's all the same story. The paper says nothing, says it's evolution. Puts a phylogenetic tree and says it's evolution. So
yeah,
at the point it's not small changes.
Right. I always ask people to show me the chemistry. I can understand the chemistry. Show me the chemistry and how this happens. No black box that this goes from this to this. Show me the chemistry on how that would occur. And the explanations go away. The papers don't show the chemistry and how this thing can occur. Because the only thing they can say chemically is that you got a point mutation in DNA, and then you got another point mutation, and another point mutation. And how many point mutations are you going to need to change one system into another? And what's the probability of something like that happening without breaking the whole system, the original system, if you're going through all of these point mutations? That's the problem. It's really getting back to the heart of the chemistry of it.
So in your opinion, is the age of the Earth, for example, enough for some randomly duplicated strand of DNA to be mutated point by point into a kinesin? Do we have enough?
Yeah, it's not a matter of opinion. This has all been calculated. So if you take the universe, if you take the scientist's number of the universe, 14 billion years, if you take the scientist's number of the age of the Earth at 4 billion years, is that enough? And the calculations themselves say it's not enough to do all of this that would have to take place. So when you look at the improbability of all of these things happening, if you take those numbers that the scientists themselves give, the mathematics is going to lose. The mathematics wins over the evolutionary process. That wins all the time. The mathematics says no, evolution says yes. So that's the problem of what we're dealing with here. I don't use the mathematical argument because others have used that. mathematicians have shown the real trouble with evolutionary suggestions. That's been shown for a long time, but for some reason they say that mathematicians just don't understand. So that's why I'm using my expertise, which is the chemistry, to say the chemistry is just not there either.